Hello and welcome. My name is Lleahy and this is my multimedia presentation on microbiology aimed at junior cycle students. The learning outcomes. At the end of this presentation, the students will be able to differentiate between viruses, bacteria and fungi. They will have investigated microorganisms present on skin, and they will be able to identify the harmful effects and diseases caused by microorganisms. The rationale for choosing this topic. There are many reasons why we are studying microbiology. Most important amongst them are understanding microbiology and microorganisms is important to human health. There are opportunities for investigations and experiments which promote the key skill of managing information and thinking. There are also many active teaching methods that can be included to encourage the key skills of communication and working with others. And there are cross-curricular links with home economics. What are microorganisms? Micro means small and organism means living thing. So if we put that together, microorganism means they are tiny living things, most of which are not seen without a microscope. They live everywhere, in the skin, soil, air and water, and inside plants, animals and humans. There are three types that we will look at: viruses, bacteria and fungi. Viruses. These are the smallest of the microorganisms. They are unable to reproduce by themselves, and they must invade other cells to live. Therefore, they are parasites. Antibiotics do not kill them. They can cause disease. Common diseases that they cause in humans are measles, mumps, chickenpox and AIDS. Influenza, commonly called the flu, is also a virus. This picture depicts the flu virus under a microscope. The next microorganism we will look at is bacteria. These are larger than viruses. They reproduce asexually, which means that new individuals are formed from only one parent. Antibiotics can kill them. They live on dead matter, which makes them a decomposer. They need four things in order to grow. They need food, water, a suitable temperature, and a suitable pH. And common examples of bacterial infections in humans are pneumonia, tuberculosis, commonly called TB, and tetanus. This is an example of microorganism growth on a hand. A hand was placed on top of a petri dish filled with nutrient agar. It was then covered and placed inside an incubator for 3 days. After that time, this is the microorganism growth that was seen. The last microorganism we look at is fungi. These are simple plants that do not have chlorophyll. Because they do not have chlorophyll, they are unable to make their own food. So some fungi are parasites and some are decomposers. Some are single-celled, making them quite simple microorganisms, and others are long threads that can live underground. Some can be eaten, for example mushrooms, and others are poisonous. Diseases they can cause in humans include athlete's foot. They also cause food to go off, for example bread mold. This is an example of bread mold caused by fungi. In the first picture, we can see a slice of bread that obviously contains bread mold. On closer inspection, we can see in the bottom picture that the bread mold consists of long, thin threads. This is the fungi that leads to bread mold. It is known as rhizopus. We will now investigate microorganisms present on a hand. You have each been given a sterile petri dish containing nutrient agar. Open the lid and touch the top of the agar with one hand or one finger. Next, close the lid of the petri dish. Label the underneath side of the dish with a marker. You can put your name and the date. One dish will remain unopened and will be the control for the investigation. We will now place all of the dishes upside down in an incubator at 25 degrees Celsius and we will check the results in 3 days. The results of the investigation. The Petri dish went into the incubator looking like the top picture. After a few days, when we check it, we would expect to see something similar to the bottom picture. The area touched by the fingers can be clearly seen due to the growth of microorganisms. Some questions now to consider in pairs. Question 1. Why did we leave one Petri dish unopened and untouched? Question 2. What would we expect to see on this Petri dish? Question 3. Why did we place the Petri dishes upside down in the incubator? And question 4. Any ideas on how we could extend, improve, or develop this investigation further? Answers. Why did we leave one Petri dish unopened and untouched? This acts as a control, and the control acts as a comparison. It is how the Petri dish behaves without contaminants when left in an incubator for 3 days. Question 2. What would we expect to see on this Petri dish? We expect to see nothing on the control, no growth of microorganisms. Question 3. Why did we place the Petri dishes upside down in the incubator? We placed them upside down because condensation from the growth of microorganisms would block our view if we did not. Question 4. Any ways we could extend, improve, or develop the investigation? We could use clean hands, dirtier hands, a school pen, or any other object that we wanted to test for the presence of microorganisms. Have the learning outcomes been achieved? Students will now be able to differentiate between viruses, bacteria, and fungi. They will have investigated the presence of microorganisms on the skin, and they will be able to identify the harmful effects and diseases caused by microorganisms.